Hi, it's Mr. Anderson, and welcome to Biology Essentials Video 35. This is on viral replication, and this is a painting of Benjamin Franklin. Benjamin Franklin was always puzzled by things that he didn't understand, and one thing he didn't understand was the common cold and what caused it. And so this is something he wrote where he's talking about what causes the cold. Traveling in our severe winters, I have suffered cold sometimes to an extremity only short of freezing. But this did not make me catch cold. I've been in the river every evening 2 or 3 hours, a fortnight together, when one should suppose I might imbibe enough of it, take cold if humidity could give it, but no such effect followed. And so what he's testing is this idea that colds are caused by being cold. And what he found, just like I did growing up, is that that's not really what causes cold. And so what he settled on was, I love the wording here, it was the frowsy, corrupt air from animal substances and the perspired matter from our bodies. And so Benjamin had settled on this idea that there was something passed between us that causes cold. Now this is before the whole germ theory, before we even identified bacteria alone viruses. He was starting to hint at this idea of a cold. What does a cold look like? Well, we now know that a cold or a rhinovirus is going to look like this. This is a picture of a typical rhinovirus. It's called rhinovirus because it's going to infect this upper portion of our respiratory tract. This is what it looks like. Inside it, there's going to be genetic material. The genetic material is injected into the cells in the upper respiratory tract, and it just hijacks them to make copies of itself, to make more of the cold virus, which we then pass on to somebody else. Now, what's interesting about rhinoviruses is that there are actually 99 different types of rhinoviruses, so almost 100 different types of rhinoviruses. Each of them are very different. So even though you might never get the same cold twice, there's still a lot of colds out there, and they're going to mutate as well. And so in this podcast, I'm going to talk about viral replication, how viruses make copies of themselves. They mainly do this through a cycle called the lytic cycle, where they actually assemble the parts of a virus, and then those will eventually leave the host cell. Two things that I'll add to that is that sometimes related viruses or viruses that are around at the same time can actually swap genetic material. And then I want to talk briefly about RNA viruses. A perfect example of that is HIV and how they actually make copies of themselves. It's a little bit different. And then finally, I'll talk about this, the lysogenic cycle. So in some viruses, they'll make copies of themselves, but also they can hide out in a lysogenic cycle where they can eventually go back to the lytic cycle. What we're finding is that when they enter into the lysogenic cycle, especially in bacteria, you can actually change the virulence of those bacteria. In other words, you can make them more or less pathogenic or dangerous. Now, this is my kids' version of the lytic cycle, or how viruses make copies of themselves. So it's not super accurate. I wish viruses look like that, but they don't. And so what do you need to be a virus? Well, you need to have some genetic material, DNA or RNA. You have to have something to put it in, so there's usually a protein coat. And then a lot of them will have membranes around the outside that allow them to fuse with a host cell better. And sometimes they'll actually take those membranes with them when they leave. So what happens in the lytic cycle is that that virus will actually drop off some of its DNA. That DNA will move into the nucleus where the machinery of the cell will make a copy of it. And so this process right here is called DNA replication. So if you're paying attention, what did I say they drop off? Well, really all they have to drop off is the genetic material. So how do they make copies of the DNA if they're just dropping off the genetic material? Well, the cell's machinery will actually do that. Watch what happens next. That DNA will make copies of itself called RNA. That's called transcription. So transcription is the creation of RNA from a DNA template. And then what happens next? Well, each of those messenger RNAs will go to a ribosome where they'll actually convert the message of the messenger RNA to proteins of the virus. So what do we call that? That's called translation. And so what they're using is using the machinery of the cell to not only duplicate the DNA, but to transcribe that into messenger RNA and then eventually to make the proteins of the virus. And so it's pretty insidious. And you can see why we don't call viruses alive, because they really don't have the machinery of a live cell. They're using the machinery of a cell to actually make copies of themselves. So let me get that out of the way. What happens next? Well, this is why we call it the lytic cycle. The lytic cycle, or to lyse, means to break. And so usually when the virus actually escapes, it'll lyse the cell, it'll destroy the cell, and it'll spread that virus out. And so what's happening is you only get a few viruses into a few cells in your upper respiratory tract. They make millions and millions of copies, which infect cells, and it goes just like a computer virus. It grows exponentially. Now, if you're paying attention, you notice somebody else snuck in here as well. So this guy looks a little bit different, but he happened to be inside the cell at the same time. So he could take some of his genetic material with him or some of this new genetic material. And so we can get variation in viruses as well. Now, some cells are actually, or some viruses are what are called RNA viruses, or sometimes we call them retroviruses. What do they have? Well, their genetic material, you can see, is not DNA. It's going to be RNA. They'll also usually have an enzyme that goes along with them. In this case, they have something called reverse transcriptase. So if you don't know what that is, let's watch what happens in the animation. So they inject RNA and reverse transcriptase in. And what does reverse transcriptase do? Well, it does the opposite of transcription. In other words, it takes RNA and actually makes DNA out of it. Now, why is that bad? Well, there's not going to be a lot of error checking within there. And so in RNA viruses, you have mutation rates that are just crazy. They're not scanning the DNA to make sure there are no mistakes. The RNA constantly has mistakes. And in fact, RNA viruses will actually live on the mistakes that are made in the copying of that. So what happens in an RNA virus, same thing as in the last slide, they'll make RNA and they'll make proteins, and then eventually that leaves on its way, along with reverse transcriptase. And so here's an example of a RNA virus. This is called HIV. And so we can see in here that they've got the RNA, so they've got this genetic material inside here, but they also have reverse transcriptase, that enzyme that looks like that. They also have proteins around the outside, and they also have a membrane around the outside. And so HIV is an example of an RNA virus. And what I said just a second ago is that RNA viruses love mistakes. They love high mutation rates. And so the problem with HIV is that we can start to make a vaccine, but they'll mutate so quickly that it's not going to work. And so RNA viruses is kind of a double-edged sword. On the one side, being RNA as their genetic material makes it harder to attack them and find medication for them. On the good side, this reverse transcriptase has been a target that we can actually use to make most of our anti-AIDS or anti-HIV drugs. And so it's kind of a different kind of a virus, mutates really, really quickly, but also it allows us to treat it as well. Last thing I want to talk about is the lysogenic cycle. And so I've kind of walked you through this, which is the lytic cycle. That's where a virus injects its DNA, it makes copies of itself, it makes the machinery of the virus, and then eventually this would be that lysine, or that release of the viruses to go infect more cells. But there's also a different cycle, and that's called the lysogenic cycle. Lysogenic means creating lytic is the best way to think about it. It can give birth to the lytic cycle. So what happens here is they'll inject their DNA in, it'll actually sit in the bacteria, and then as the bacteria copies itself, it'll be copied over and over and over again. So that's pretty cool. In the lysogenic cycle, which is not in all viruses, they're actually getting the bacteria to copy themselves over and over and over again. They can eventually jump back into the lytic cycle, and then this continues. So cold sores are an example of that. If you have cold sores, or those big sores on the outside of your lip, you'll have those forever. You have the DNA inside the cells inside your body. They're sitting in the lysogenic cycle, and then they can jump into the lytic cycle when your immune system is suppressed. So it's pretty cool, at least cool from the side of the virus way to pass themselves. But the other thing that's interesting about the lysogenic cycle is that when bacteriophages, and bacteriophages, remember, are going to be viruses that infect bacteria, when they enter into the lysogenic cycle, they can actually change the virulence of the bacteria. And so cholera is an awful disease. It's a diarrheal disease. So you die as a person with cholera. You die of severe dehydration. It's caused by a bacteria, Vibrio cholerae, and it's been around for thousands and thousands of years. But what we've discovered is that cholera itself is actually not that bad. It's not very virulent. It won't give off toxins until it's infected by this virus. And once it's infected by this specific CTX virus, then it becomes virulent and can lead to these awful cholera outbreaks. What we find, though, is that natural selection is not always our enemy. And in cholera updates, if we can clean up the water supply, we can actually force the cholera bacteria to evolve less tolerance, a tolerance where they don't have the phages and they can't pass them on. And so we're actually using that for good. And so that's how viruses make copies of themselves. Remember, all they have to have is a little bit of genetic material, a protein to carry them in, maybe a membrane, and they use the machinery of us to copy themselves. And so those are viruses, and I hope that's helpful.